the lectures that focused on alleles. So the alternative forms of the gene at the same locus called alleles. For example, as you can see here in the pictures below, this is our gene of interest, and each gene located located on allele, which is the form of the gene. So we will inshallah discuss on the next slide the status of this gene, if they are the same, identical, or if they are different. But here now just remember that the alternative form of the gene at the same locus, at the same location, are called allele. For example, the ABO, if we are interested in the ABO gene here, there is three main allele: A, B, or O. So you can inherit A from your mom, you can inherit B, you can inherit O. The same from your dad, you can inherit A, you can inherit B, and you can inherit O. A blood group system. Now, you understand what is the carrier, you understand what is the gene, the antigen, what is the gene, what is the allele. The blood group system is defined as the collection of the antigen controlled by allele at the single genetic locus. For example, the ABO system. So there is a two option. Either I have only one gene that encoded the carrier, or I have multiple genes, they sit close to each other and they can encode the carrier. So as an example of the single genetic locus, the allele at the single genetic locus, for example the ABO system, or two or more closely linked loci as seen on the Rh system, Rhd gene and Rhce gene. Allelic status. The homozygotes, an individual who inherited the identical allele from each parent. For example, I get one allele from mom, one allele from dad. In this case, we call them homozygotes allele. And I have the A blood group. If I inherited B from mom and B from dad, then I have a homozygotes allele and my blood group will be B. The same with the O. As you can see here on the pictures, the left, the circles on the left, on the right, I mean. So both the gene B capital or both gene or both allele is the B small. In this case, we call it homozygotes. For the heterozygotes, an individual who inherited different allele from each parent. For example, I get A from my dad and O from my mom. So they are different, is the heterozygotes for that gene. As you can see here, the circles on the left, it's specified by the color. So it's pink and yellow, or yellow or small b or big B. So they are different form of the gene, different allele, and we called it heterozygotes. Okay, the impact of the zygosity on the antigen expression. The zygosity effect, we called it dosage effect on the blood bank on the serological test. The variation on the strength of the antibody reaction depend on the whether a red cell's antigen is inherited in the single dose heterozygotes or double dose homozygotes. The homozygote cell with the higher antigen density usually reacted more strongly than a heterozygote cell. وبالتالي thus we can set this like simple equation, the homozygotes, if I have a homozygotes allele, that means I have a double dose of the antigen. This will give me a stronger reaction with the antibody. If I have a heterozygotes, different form of the allele, I get a single dose of the antigen. That means I will have a weaker reaction with the antibody. Next slide have an example so you can understand this more quickly. So the example here from the MNS system, the MN antigen are allele at the same locus, as you can see here on the pictures. I will talk now on the pictures on the left. So in one allele I have M, in one allele at the second allele I have M. So in this case I have a homozygotes allele. That means they can encode the M antigen. That means I have dense M antigen on the red blood cell. The same if I have here, instead of M, I have N, and the second allele have N. They will encode the N antigen. So I have the background, the genotype here, M or N. That means I will have this phenotype. This is the way we wrote the phenotype. We write the phenotype. So if it's the homozygotes for M, it's the M plus N minus. If it's a homozygotes for the N, the M minus N plus. So this is the genotype, this is the phenotype. We will talk more about it inshallah on the next slides. So what's the result? I will get a strong agglutination because the antigen is expressed on the double dose. As you can see, because I have M allele on the same locus, MM, we call it double dose. Okay, and the both allele will encode the same antigen, which is M. Or they can, in case of the N, I will have two allele encoding N, so I will have a dense N antigen on the red blood cell. This is in regarding to the homozygotes. In case of the heterozygotes, as you can see here on the pictures inside the red rectangular, I have the M on the one allele and N on the other allele. So I have the heterozygotes allele. That means I have a single dose of M and the single dose of the N antigen on the red blood cell. And this is how we wrote, how we write the phenotype, M plus N plus. The result, I will have a weaker agglutination because each antigen is expressed in only single dose. So if I have, if the patient have the anti-M, it will react with the red cells having the M single dose on them, and they give the weaker reaction comparing to the homozygotes. M and N, because they are located on the same gene locus and they are different allele, we call them on the red blood cell antithetical antigen. Antithetical mean they are opposite, but the very important thing, they should located on the same location on the chromosome. So the antigen encoded by the allele at the same locus are called antithetical opposite antigen encoded by the allelic gene. For example, for the KEL system, so the previous slide, we have the MNS system, the M and N, they are located in the same locus, they are antithetical antigen. The same for the KEL system, we have the KEL, the big K and the small k, they are antithetical antigen. So it is wrong to say that the K minus K positive, which is the phenotype, is the homozygotes for K. The correct way to say it is the person is the homozygotes for the allele that encode K, small k, giving a double dose of the K antigen. Some hospital they called the small k Chilano. So in this case, the person is the homozygotes for the allele that encodes Chilano, giving a double dose of the Chilano antigen on the red blood cell. Another terminology, genetic terminology, the haplotype. If you look, let's see the example first, then we can understand the definition. So on the chromosomes 1, the Rh gene or genes located on locus 1, RHD, and locus 2, RHCE. They are close from each other and they tend to be inherited together. So for example, if I RHD, if I want to give my son or my daughter RHD, the RHCE travel with it. In this case, we call them a haplotype. So a group of allele that tend to be inherited together are referred to as a haplotype. RHD and RHCE allele on the Rh locus are the example of the haplotype. Now we're going to talk about the phenotype and the genotype on the blood group system. The phenotype, the observable trait, what is expressed. So for example, the color of the eyes, the color of the skin, the nature of the hair. And on a blood group, the phenotype refers to the presence or absence of antigen on the red blood cell, which is detected by the serological testing. Example, testing with the ABO system, testing with A and B reagent. So I have the anti-A and I have anti-B, they come from the company, and I test them with red blood cell individual, either donor or recipient. Okay, let's go to this picture. Maybe it's or we can read and refer to the picture at the same time. If the agglutination happen with anti-A, so see in the pictures, the first row, I have I put on the slide 2 drop of the blood. One I tested with the anti-A from the company, and the other one I tested with the anti-B. I get the agglutination with the anti-A, that means this patient have the A antigen on the surface of the red blood cell, that means the blood group is A. The same with the B. If I get the agglutination with the anti-B, that means anti-B reacted with the B antigen and give me the agglutination or reaction. That means the person is a group P. If there is a reaction in with the A, anti-A, and with the anti-B, that means the persons have both A and B antigen, and the phenotype is AB blood group. If I don't get any reaction, that means the patient do not have any antigen on the red blood cell, and in this case, the blood group is O. Because I can see it with my naked eye, this is a phenotype. Okay? What is the genotype? The actual genetic makeup, the inherited alleles, we call it genotype. For example, of a person with the phenotype A, I see it on the lab that the blood group is A, could have a genotype AA or AO. Only molecular testing or family study can distinguish. So either I test the allele and see what the allele the person's inherited from his mom or his dad, or I can track the phenotype through the family. What's the blood group of mom? What's the blood group of dad? What is the blood group of the sisters and brothers, to be able to distinguish between this two genotype makeup. The O gene produces no antigen. So if the persons inherited A from mom and O from dad, appear phenotypically the same as AA. We will talk about this situation inshallah on the inherited section. Two people, that mean two people with a phenotype A may carry different genotype. What's the polymorphism? We talked about there is a multiple form, or we called alternative form of the gene allele. So the polymorphism is having two or more allele at the given genetic locus. For example, the ABO system has a three allele, A, B, O. That means it is intermediate polymorphic gene. Why we care about the polymorphism? Because its impact on the population. How? We will see. The degree of the polymorphism affect the frequency of the phenotype on the population. So for example, if we, for example, this population have 90%, for example, of the A blood group, that means because I have a two form of allele can produce this phenotype, I can have AA or AO that that they are able to give me an A phenotype, and this is give me how frequent this phenotype in the population. The degree of the polymorphism, how many allele I have, affect the frequency of the phenotype on the population. A highly polymorphic system make it less likely to find two people with the same phenotype. So some blood group systems are more polymorphic than others. For example, ABO, moderately polymorphic. Why? Because they have just three main allele. They have many variants, and we will talk about the variant later, but they only have a three main allele. RH is a highly polymorphic because it has hundreds of alleles. We may just stop here and continue with our third part when we talk about the genetic variation and mutation on the blood group system.